Слышно нас? Скажите, пожалуйста, те, которые в Zoom.
Здесь слышно. Сіздер біз естіп тұрмыз.
Иә, жақсы естіліп тұр, сәлеметсіздер ме?
Штурк. Балаға кірді.
Hello, Railam. Hello, everyone.
How are you?
Very
Muhammad.
well. How are you, my dear brother, Professor Adikinov? So happy to see you.
To you.
Thank you, my dear. Thank you. Thank you, Zuldos. Thank you. I see all friends, Bulat is here. Assalamu alaikum, my dear brother.
Jackie, assalamu alaikum. Jackie, assalamu alaikum.
I wish I can come right away to you. Railam, can you translate that? I am just so missing so much my brother Abilov that I wish to come soon, inshallah.
Okay, thank you. Alex is anyone? Alex is anyone shut the function for this event? And he's also happy to see you. Thank you so much.
Thank you. Thank you to the panel of referees and examiners. Thank you.
Ссылка не открывается у него. Мехмед Устурк. Вроде здесь. Не знаю. Сейчас
Саламатсыздар ма? Good afternoon, dear colleagues.
How are you?
Thank you, fine.
Selam Aziz Bey.
Walaikum salam, brother Mehmet. How are you?
Professor Chaudhry, how are you?
Very well. Good to see you.
Yeah,
After a long time.
I am very delighted to see you.
Absolutely. Thank you so much. Thank you.
Thank you so much.
Давайте я вам отправлю еще раз.
апаймен сізге жіберген ссылканы жіберіңіз, сол жоқ.
Well, hello, how are you? Merhaba. Amanzholkyzy.
Идентификатор код бар ма соны мына жақта көрініп тұрған жоқ. Апай, мен сізге жібердім идентификатор кодты.
How to start? We would be starting. What is the agenda for today? Who's supposed to speak first? I'm sorry, I don't have the agenda. Mehmet, can you? Yeah.
Agenda is like 20 minutes we will talk for applicant, and after that discussion, and after that the local and foreign supervisors will give a talk, and after that reviews will give their opinion and discussion, and after that vote.
Да, добрый день, коллеги.
The defense of the dissertation by Amanzholkyzy aralarım. On the topic, Israeli also federal review materials and britannically based on random standard analysis speed and especially 805301 chemistry. Scientific supervisor Sabino Shafasnovich is here. Doctor of Chemical Sciences, Professor of Chemistry and Technology for Organic Substances, Natural Compounds and Polymers, and Parabic as National University. For me, supervised with Amanzholkyzy, Doctor Professor, Medical General Conspect, Distinguished National Professor, Honorary Advisor, ICCBS, Karachi, Pakistan. The dissertation was performed at the Department of Chemistry and Technology of Organic Substances, Natural Compounds and Polymers, at the Research Institute of New Chemical Technologies and Materials of the Parabic National University. Official reviews at the Harvard candidate of Chemical Science, Associated Professor, Deputy Head of the Laboratory of Engineering, of the Satpayev University of Kazakhstan. And проживает в Иван, Doctor of Chemical Sciences, PhD, Professor Buket of Karaganda University, Karaganda, Kazakhstan. Temporary members of the Dissertation Council, Oztur Mehmet, PhD, Professor Dr. Mugana Sikir, Oman University from Turkey, Adil Molvir Jamshayevich, Doctor of Chemical Sciences, Professor of Satpayev University of Kazakhstan, and Atikenov Sergazim Bajasarovich, Doctor of Chemical Sciences Professor, Research and Production Center for Chemistry, Chairman of the board. The floor is given to the Secretary, Professor Bulat Malbekov, to present the documents available in the applicant's personal file.
Dear Dissertation Council, dear reviewers, meeting, give me let me to introduce all the documents we have here in our Dissertation Council and also information about the applicant. The applicant name Amanzholkyzy, born in 1995. He took undergraduate studies at our university at University from 2014 to 2017 at Faculty of Chemistry and Chemical Technology, a major in chemistry. In 2017, she successfully enrolled to master program at Farad University, specialty in chemistry, and successfully completed this study in 2019. From September 2020 to May 2014, I'm talking update your PhD. Enrolled to PhD program at Faculty of Chemistry and Chemical Technology under the educational program in chemistry. The topic of her dissertation and her advisors are by Academic Council of Kazakhstan University on November 2, 2021. Also, the correction was done of her topic on July 8, 2025. The personal file, the personal documents includes her bachelor and master diplomas, list and the copies of her publication, positive recommendation. Dear colleagues, hear me?
Уважаемые коллеги, вы слышите?
Слышно нас, уважаемые коллеги?
Микрофонға жақын сөйлесеңіз жақсы болар еді. Дауыстарыңыз алыстан естіліп тұр.
А сейчас попробуйте.
Себя слышите?
Слышно, но только плохо слышно.
Dear colleagues, we have some problems here, technical problems. Just a moment.
Dear colleagues, do you hear me? Слышно меня?
Ya bu mikrofon жақсы болады.
Жақсы естіледі, сіздер микрофонды жақын ұстасаңыздар. Алыс тұрасыздар, сол кезде естілмей қалады.
Булат младший, нас слышно, да? Теперь лучше?
Да, теперь намного лучше.
Да, лучше.
Намного лучше.
Я хотел спросить, до этого, когда представлял председатель, это было слышно или ничего не слышно, придется заново записать?
Нет, не было слышно.
Хорошо, сейчас заново начнем, пожалуйста.
Okay. Today's defense of the dissertation by Amanzholkyzy on the topic hydrogel forms of hydropropyl methyl cellulose and bentonite clay based on plant extract Amarix hispida. Specialty 8D 05301 chemistry. Scientific supervisors Abilov Жарылқасын Abdohitович, Doctor of Chemical Sciences, Professor of the Chemistry and Technology of Organic Substances, Natural Compounds and Polymers, Al-Farabi Kazakh National University. Iqbal Chaudhary, Doctor, Professor, Coordinator, General Comstech, Distinguished National Professor, Honorary Advisor, ICCBS, Karachi, Pakistan. The dissertation was performed at the Department of Chemistry and Technology of Organic Substances, Natural Compounds and Polymers, and the Research Institute of New Technologies and Materials of the Al-Farabi Kazakh National University. Official reviewers Tatykhanova Gulnur Sayranovna, candidate of Chemical Sciences, Associated Professor, Deputy Head of the Laboratory of Engineering Profile of the Satpayev University, Almaty, Kazakhstan. Tajibbayev Yerkibulan, Doctor of Chemical Sciences, PhD, Professor Buketov Karaganda University, Karaganda, Kazakhstan. And temporary members of the Dissertation Council, Astürk Mehmet, PhD, Professor Dr. Muglasitki, Folkman University, Turkey, and Abdiyev Kaldыбek Jamshavich, Doctor of Chemical Sciences, Professor Satpayev University, Almaty, Kazakhstan. Adikenov Serghazy Manjasarovich, Doctor of Chemical Sciences, Professor, JCS Research and Production Center, Phytochemistry, Chairman on the board. The floor is given to the Scientific Secretary, Professor Bulat Uralbekov, to present the documents available in the applicant's personal file.
Yeah, hello, dear colleagues, finally, and dear dissertation council members, reviewers, the community, and let me to introduce information about Amanzholkyzy Araylem and all the documents we have in the dissertation council. Amanzholkyzy Araylem, born in 1995. She enrolled to graduation studies at Al-Farabi Kazakh National University from 2013 to 2017 at Faculty of Chemistry and Chemical Technology, majoring chemistry. Yeah, in 2017 she successfully enrolled to master degree at Kazakh National University, specializing in chemistry, and completed her studies in 2019. From September 2020 to May 2023, Amanzholkyzy Aralım enrolled to her PhD program at our Faculty of Chemistry and Chemical Technology under educational program Chemistry. Her topic of dissertation and advisors were approved by Academic Council of Kazakh National University on November 2021, and it was done some correction to her topic. Yeah, and it was done some correction for her topic on July 8, 2025. The personal files includes copies of her bachelor and master diplomas, list of copies of her
Не слышно вас?
Ваш микрофон отключили кто-то.
yeah. Just a okay. And as a part of her dissertation research, the doctoral candidate published 8 studies, including 1 papers in journal Химия растительного сырья, indexed in Scopus, and 1 paper in journal Molecules, indexed in Scopus and Web of Science, and also 1 patent for utility model. Also 3 abstracts presented in international conferences. On September 2, 2025, Amanzholkyzy RLM dissertation work was discussed at an expanded meeting of Department of Organic Substances, Natural Compounds and Polymers at the Faculty of Chemistry and Chemical Technology. And the meeting was 30 participants attended this meeting. The first reviewer, candidate of Chemical Science, Associate Professor of the Department of Chemical and Biochemical Engineering of the Geology and Oil Gas Business Institute, named after Tursunov, Mangazbaeva Ravasha Mantayevna. And the second reviewer was a candidate of Chemical Science, Associate Professor of our department, Eskalieva Balkhaskum professor, yeah. And all the questions and comments raised by reviewers received comprehensive responses. So the academic seminar recommended the dissertation for further defense. And this protocol was approved by board member and vice rector for scientific and innovative activities of Kazakh National University by Ibrahimov Margulan Kasenovich. And on October 8 and 10, 2025, by protocol number 4544, the dissertation council scheduled the defense date, appointed official reviewers, and also appointed designated temporary council members. So that's all. I finished.
Okay, thank you, Muratovich. Are there any questions for the scientific secretary or PhD candidate? Do you have any questions, dear colleagues? If there are no, the floor is given to the applicant to present the content of the dissertation, and Aralım, you have 20 minutes. Okay, please.
Good afternoon. Do you hear me?
Yes, it's clear.
Okay. Today's dissertation thesis theme is hydrogel forms of hydroxypropylmethylcellulose and bentonite clay based on plant extract Tamarix hispida. So cellulose derivatives, particularly cellulose ethers, are rightfully considered the leading natural materials for hydrogels as drug delivery systems. These systems are easy to use, painless, and provides increased efficiency due to the prolonged release of the active ingredients. Hydrogels also functions as a matrices in which the biological active compounds, synthetic or natural plants, as well as plants materials, can be immobilized for biomedical applications and to enhance the biocompatibility. Among the cellulose derivatives, the most widely used are methylcellulose, hydroxymethylcellulose, hydroxyethylcellulose, and hydroxypropylmethylcellulose. To improve the properties of the natural polymers, particularly cellulose derivatives, composite materials are often developed in which the mineral components serves as fillers. One important and compatible and structure reinforcing component for the polymer composite is an inorganic mineral, bentonite clay, and the large deposits and the reserves of bentonite in Kazakhstan is an additional incentive for development of organomineral hydrogels. The aim of the dissertation is to obtain new hydrogel forms based on hydroxypropylmethylcellulose, bentonite clay, and the extract from the aerial part of the Tamarix hispida plant. And to reach this goal, the following 4 tasks was formulated. The main provisions for the dissertation work is: first, the optimal conditions for obtaining a water alcohol extract from the plant extract Tamarix hispida by ultrasonic extraction at time of extraction 25 minutes, a raw material to solvent ratio of 1:6, a temperature of 20 till 40 degrees, and an ultrasound frequency of 35 kilohertz, which ensure the highest yield of biological active compounds. The second is aqueous ethanol extract from the aerial part of Tamarix hispida, demonstrates pronounced anti-inflammatory and antioxidant activity. The THU10 extract demonstrates a similar level of DPPH radical inhibition with gallic acid and quercetin standards at concentrations of 400 microgram per milliliter and higher. Based on the third is based on the analysis of rheological properties, the optimal hydrogel compositions: a hydrogel based on bentonite clay 25%, a hydrogel based on HPMC 2 till 4%, a composite hydrogel based on HPMC bentonite clay 4%, 7:3 ratio, with addition of 1% THU10 extract. The main objects of the research is the plant raw material and its extract, bentonite clay, and hydroxypropylmethylcellulose. The Tamarix hispida plant genus is the family of Tamaricaceae, is a valuable source of the biological active compounds. The terpenoids, polyphenolic, and other biological active compounds was previously isolated and characterized, and the biological activities such as antimicrobial, antifungal, and etc. Was previously founded. And the bentonite clay was collected from the Manrak deposit, and the bentonite clay was purified by the several precipitation in the distillate water, and the content of the bentonite clay is up to 70% of montmorillonite, which is good for use in medicine. Also, for extraction of Tamarix hispida plant, we used ultrasonic extraction method because ultrasonic extraction method is a faster method for than the traditional methods like maceration. The Tamarix hispida plant was collected at the flowering period and collected and then drying and grinding. The grinded plant material then given for ultrasonic treatment for 25 minutes, about 40 degrees. Then it's concentrated and dried in the rotary evaporator. The extract was dark brown colored, and it has specific taste and smell. Then this extract powder are given in the for qualitative and quantitative analysis, and also for biological activity analysis. Also for the Tamaris hispida extracts, we made two types of extraction: is the traditional maceration extraction and the ultrasound extraction. As it's shown in the table, the extract of THU10 showed increase in the extractive substances yield. It's increased up to 37.33 or 34%, and also we can see 2.7-fold increase in the flavonoid content in the Tamaris hispida ultrasound extract of which is obtained by ultrasonic method. Also, the quality indicators such as humidity, ash content, extractive substances, tannins content, and flavonoid content was found. And for the next works, the THU10 extract was used for immobilization for carriers, for polymer carriers. Also, the optimal conditions for ultrasound treatment extraction was found, and the optimal compositions for the THU10 were determined as follow. Raw material to solvent ratio is 1:6. Extraction duration is 25 minutes at the ultrasonic frequency of 35 kilohertz, which at these parameters we can see the increase of yield of extractive substances up to 37.34%. And as using this ultrasonic method, we can see the yield of extractive substances increased by 3 or 4 times, and it significantly reduces the time of extraction from 48 hours to 30 minutes. Next, the phenolic composition of the extracts THU10 5070 was found by HPLC. In the table, we can see the THU10 extract is content has a gallic acid, 4-hydroxybenzaldehyde, epicatechin, rutin, fisetin, and some fumaric acid and hesperidine trace amount. The THU50 extract also has these compounds, but in another amounts. But there we can see the absence of the gallic acid and some fumaric acid. And the THU70 extract also has protocatechic acid, also vanillic acid, which is absent in previous extracts, and some ferulic acid, fisetin, and rutin extracts. It was identified the biological activity of the anti-inflammatory and cytotoxic activities of this extract. It was identified by the HRBC membrane stabilization method. For this method, the volunteer was taken the blood from the volunteer who is not taken non-steroidal anti-inflammatory drugs for 2 weeks, and the blood was centrifuged and added Elsevier reagent, and then the serum was identified spectrophotometrically. So all biological activities were investigated in the laboratories of HEJ, ICCBS at the University of Karachi, Pakistan. And as we can see at the table, the anti-inflammatory activity is in the range of 32 and 70%. And all 3 extracts didn't show the cytotoxicity, which is good for using them as a biological active substances for human organisms. And we can see, we tested the solubility in water, and we can see that THU10 have a good solubility, but the 50 and 70 is partially soluble. Also, antioxidant activity was evaluated by the DPPH method. The antioxidant activity of studied THU sample was compared with reference antioxidant standards such as quercetin and gallic acid. When compared with the quercetin and gallic acid, the THU10 extract demonstrates a similar level of DPPH radical inhibition at concentration of 400 micrograms per milliliter and higher. Also, the THU10 extracts are given for VLC fractionation. VLC fractionation was subjected using silica gel as a stationary phase and eluted via gradient mix elution system by increasing the polarity. And all the fractions are named and weighted and tested for solubility. Then these fractions given for anti-inflammatory biological activities. As we see in the table 6, anti-inflammatory activities of THU10 fractions was tested at the concentration of 50 microgram per milliliter and 100 microgram per milliliter, and almost all fractions showed anti-inflammatory inhibition. But the fractions number 2, especially 3 and 4, showed a high inhibition percent of the anti-inflammatory activity. So these 3 fractions are then given for LCMS analysis. Here we can see the results of LCMS analysis, and these 3 fractions has some substances, organic substances such as tripeptides, alcohols, organic acids, flavonoids, and the presence of isoliquiditigenin, 3,5-dihydroxyflavone, 3,4-methoxyphenyl, and 4-methylcoumarin, normal propyl gallate. These substances, by the literature review, are responsible for the anti-inflammatory and antioxidant activities. Then we studied bentonite clay compositions with the plant extract of THU10, and to evaluate the properties of the compositions, the parameters such as degree of swelling, sedimentation, SEM, and X-ray analysis was evaluated for these composites, as we see in the figure 8. Here we can see the degree of swelling of bentonite in aqueous, in water solution, in saline solution, and in the solutions of THU at the different concentration. As the concentration of the THU10 increases, the degree of swelling decreases, and the 1% of 1% of THU10 also comparable with the water and have a significant differences. Also, the same images for bentonite clay before immobilization and after immobilization of the THU10 was given in the figures, and also the IR spectra of the pure bentonite and extract and the composite of them are given in the table. Here we can see the specific bands for all 3. Here for bentonite clay, the specific band is 914. This is aluminum hydroxide, aluminum spectrus, and for THU is the hydroxyl group of flavonoids, tannins, and in composite we can see the appearing of the spectra band at 467 aluminum O and the silicium O. Also, we studied some X-ray structural analysis. Here we can see that the Tamarix hispidum is not only sorbet on the surface of the bentonite. Also, we can see that the sorption and the complex is appearing between the layers of the bentonite clay, and because the distance in the bentonite clay, pure bentonite clay, is 1.3 nanometers, and after the sorption of THU10, it's increasing to 1.48 nanometers. Also, the kinetics of THU release from the bentonite clay into water and into saline solution as determined. And we can see that the release kinetics of release of THU in water is higher than the into the saline solution. In 1 hour, the release of the THU increased to 58, as in saline solution, it was only 42%. Also, the rheological indicators are evaluated by the Anton rheometer, and we can see that as the percentage of bentonite clay increasing, the rheological properties are also increasing. Also, we prepared some composite hydrogels based on hydroxypropyl methyl cellulose and with the extract of THU10. The method was used hot-cold method, and for extracts also we used hot-cold method. And this hydrogels was tested for rheology, and the graph shows that the properties of the hydrogels is non-Newtonian and the non-linear nature is visible. And if the viscosity decreases, as the shear rate also increases. When the shear rate reaches high values, the polymer systems begins to flow. Also, we tested the release kinetics of THU10 from HPMC hydrogels at the different concentrations of HPMC, and the concentration of THU10 is remaining 1%. We tested the yield of released THU10 from gels in water and also in saline solutions. Also, we obtained composite hydrogels based on hydroxypropyl methyl cellulose, bentonite clay, and the extract of THU10. In the figures, we can see the composite gel itself, and also the rheological properties of gel is tested. Also, the IR spectroscopy of the hydrogels based on bentonite clay and HPMC was evaluated, and we can see in HPMC here the specific bands at the 1649. Here is the absorbed water, and also the specific band at 1108 is the ether bonds and the polysaccharide chain. And at the composite material here, we can see the specific band and 950 reverse centimeter, where we can see the presence of aluminum hydroxide bonds and some aliphatic bonds. Also the composite materials was tested for rat-paw edema animal studies. The rat-paw edema animal studies was investigated in the laboratory of HEJ of ICBS in Karachi in Pakistan, and as we can see, the THU10 exhibits anti-inflammatory activity as indomethacin standards. By this, we can, following the conclusions, aqueous alcoholic extracts were obtained from the aerial part of Tamarix hispidum plant materials by ultrasonic treatment. The optimal conditions for the ultrasonic extraction methods was determined, and after optimizing the extraction methods, the use of ultrasound resulted in a 3-4-fold increase in the yield of components from the plant material and significant reduction in extraction time from 48 hours to 30 minutes. The qualitative and quantitative compositions were obtained, extracts were determined. Also, here we determined anti-inflammatory and antioxidant activities of the studied extract, and the hydrogels based on hydroxypropyl methyl cellulose, bentonite clay, and the extract was obtained and characterized by the rheological characteristics, and were selected optimal compositions for them. Here we can see the publications regarding to the dissertation, approbation of the work, and I'm expressing sincere gratitude for my scientific advisors, for Professor Abil Abdulhitovich, Professor Muhammad Iqbal Chowdhury, for the opportunity to carry out this research. And also I'm want to express my deepest gratitude to the faculty and the department, and for my relatives, parents, and for my colleagues and friends. Thank you for your attention, and I'm ready for answering for your questions. Thank you.
Thank you, Aurelio. The presentation is complete. Dear colleagues, are there any questions? You can ask to Aurelio. Please.
I have one question.
Yes, please.
What is the maximum drug concentration, drug content in the hydrogel?
I'm not sure who asked me, but thank you for your question. We tested different concentrations, but the optimal concentration for extract THU10 was 1%, because 1% of extract is enough to exhibit the anti-inflammatory and antioxidant activities.
Do you think this is enough? Okay.
Do you have any questions, colleagues? Сұрақтарыңыз бар ма? Әріптестер, сұрақ қойсаңыздар болады кандидатқа.
Dear colleagues, dear Professor Mehmet Öztürk, it's now time for questions. So we need to ask if you have some question regarding the disease and work, so you can ask.
Congratulations, Aray. I have listened a good, wonderful thesis. And what is the next? What is your next plan in your academic career after finished PhD?
Good day, Dr. Mehmet Öztürk. Professor Mehmet Öztürk, I'm sorry. Thank you so much. Now, as a prolongation of this work, I have my project named Just Halam by the Ministry of Higher Education, by the Ministry of Science and Higher Education, and I'm planning to continue my work by obtaining another types of forms, medical forms, and testing them to use as prolonged drug delivery systems. Maybe in future also we can use some nanoparticles for prolonged and targeted drug delivery development for targeted drug delivery systems.
Okay. Okay, all right. This is you use the bentonite clay with HPMC with hydroxypropyl methylcellulose. Why you mix them? What is the benefits of mixture?
Thank you for your question. Hydroxypropyl methylcellulose is a good drug base, and to improve their properties with mineral natural compounds. As a filler, we used bentonite clay because bentonite clay have a good sorption properties, and in mixture with the HPMC, it has a good prolonged effect, and it's very convenient for drug release as a prolonged drug systems.
Okay, thank you. Thank you. I have no
Thank
more
you.
questions. Thank you so much.
Yes, please.
Thank you, Doctor.
Thank you. I have no more questions. Thank you so much, Aray. Seni
you.
kutlarım. Hı?
Dear colleagues, you can ask. Әріптестер, сұрақ қойсаңыздар болады.
Sure, I have a few questions actually. Regarding the slide number 17 or 18, I don't remember, you talked about the formation of complexes such as like this here between matrix and yeah, here. Could you once more explain these results? You said that some complexes are formed or something like this?
Yeah. As we know, the bentonite clay has a layered character. So there are some layers, and when we immobilize the THU extract, we can see that in the same images, we can see the aggregation of particles of the bentonite clay. And also after the X-ray analysis, structure analysis, we can see that the distance between these layers are increased. That means that extracts impact to the between layers distance because of the complex of THU10 and bentonite appears.
I actually agree that it's impact probably, but I don't agree that it's form some complexes. To prove that complexes is formed, we need probably to do some other analysis as well, because here based on SEM we see only some aggregation and only. Okay, the another question which I have is. Do you think that the surface area of the pentanates will somehow affect the absorption of the pH? I mean, you measured some properties like bed properties of the solvents.
The bentonite clay has already some works on bentonite clay at our department, like professors Abilov Shalkhsnab Dohitich, Shumaglieva Shnamnulan has already tested such analysis, and the bentonite clay is a good adsorbent. It can absorb like twice or thrice more mass of water than its weight. So it can be tested before, yeah. It has good adsorption properties.
Yeah, it's probably tested before, but you need to characterize your sample properly. I mean, that's why I'm asking if you measured surface area of your sorbents and if this surface area of the sorbents can affect as well the formation of gel you studied. So this was a question. Yeah, this question I have only.
Okay, thank you for your questions.
Can I have a question?
Yes, please.
Thank you. So I have a question about the chromatographic analysis which you made in your dissertation. I see that you use LCMS, but I don't see what type of LCMS you applied for analysis. Especially what type of mass spectrometer?
Here we use quadrupole LCMS with the detector of quadrupole ionization type. It seems, yeah, it's not shown in the presentation.
Yeah, but quadrupole is not an ionization technique. It's a separation technique.
Oh, yeah, I mean separation.
What type of ionization did you use?
It's written in the dissertation work. If I'm not mistaken, it was used mass ionic ionization. I forgot. I'm sorry. It's
In your
written in
dissertation, it's not provided. That's why I'm asking. So it's not provided completely. No type of mass analyzer, no type of ionization technique. I'm asking because for me it's unclear how you identify these compounds. You provide a table, okay, you say, okay, this is this compound, but how exactly did you identify? So you did the analysis, but then what did you do?
we given 1 milligram of the extract or the fractions for the LCMS, and then we added some methanol and provided some LCMS analysis. And here we can we get the library with the spectrum, chromatogram, and some specters M to Z. And then by these chromatograms, we found, by these masses to Z, we found these compounds in these fractions.
What type of spectra did you get? How did they look, the spectra? Can you show the spectra? Because the dissertation is not provided.
It's not provided. Yeah, we have spectrums, but we didn't add it in the dissertation.
Did you use any library?
Yes, we used NIST library, if I'm not mistaken. Yeah, this library shows us this content of compounds.
Yeah, but you said you used quadrupole mass analyzer, but quadrupole mass analyzer gives only molecular ions. So it doesn't give any fragmentation. Yeah, so without
Yes.
fragmentation, you cannot determine the structure. And with the quadrupole mass analyzer, you don't have an accurate mass to determine the molecular formula. Because for this particular mass, you can have a lot of candidates, a lot of isomers. Yeah, so
Yes,
but can you explain it?
we got the fraction, got the word fractions, and this we saw by the probability of these compounds, and the probability which is more than 7 is we took the candidates which is has more than 70-80% of probabilities.
No, it doesn't work like that. You said you use quadrupole mass analyzer, not tandem. So if you use tandem, maybe you can have some fragments, some fragmentation in the collision cell, but you didn't mention that. In the single quadrupole, you will not have any fragments, no match library, so you will have only molecular ion. Yeah, so no fragments. So you cannot identify the structure at all.
Yeah, I'm sorry, maybe I'm mistaken. Maybe we used the tandem one because we had the fragments, and by the library we found these compounds.
Yeah, but who did this analysis?
This analysis was made at the ICCBS laboratories in Pakistan.
So you didn't participate in the analysis and the processing of these results?
And I dissolved my samples. I prepared my samples, dissolved in methanol, and given for analysis. And then I worked with the library by myself.
Yeah, but it's really for me it's really confusing. Yeah, and because this you don't know these details, what type of mass analyzer, what type of ionization, whether you had collision cell or not, and this data is not provided. So for me, it's still unclear. Yeah, but don't you have chromatograms on in the dissertation? Chromatograms, mass spectra.
Yeah, it's not provided. Yeah, but we just provided in the table and showed some components which is taken from the library.
Okay, so you cannot prove that you get this particular result. There is no evidence, as I see.
Yeah, if it's needed, I can later show the chromatograms and the fragments.
But I see that you provided some HPLC with diode array detector chromatograms.
Yes,
Figure
for
21, 22.
yeah, for phenolic compounds profile, for identification of phenolic profile.
Okay, and how did you identify these peaks?
Here, in the dissertation work, here you can see the link for the article. Here, all the standards methods was used. Here's standards of 25 phenolic compounds. Did the calibration curve, make the calibration curve, and by these standards, we tested our extracts THU10, 50, and 70. And here we can see the qualitative compound of the extracts.
Yeah, but some peaks I don't, as I see, they are not separated very well. So there is a like big hump, and a lot of peaks are overlapping. So how can you be sure in the identification results and also quantification?
Yeah, by retention time and the area of the peaks, we identified these compounds.
No, but at the same retention time, you had many other peaks. You see, it's really a big hump between the 25 and 35 minutes. There are a lot of peaks. They are overlapped. So I really have doubts whether you have these particular compounds. Because like peaks are very small.
Yes, by these peaks we identified this phenolic composition, but it seems to be the resolution is not so good. It will be very good to have a better one. But this was enough for us to show that these compounds are responsible for the anti-inflammatory and antioxidant activities.
So what compounds are responsible?
For
this
example, the
activity?
gallic acid is responsible for antioxidant activities. Epicatechin, rutin, fisetin, such phenolic compounds are always responsible for antioxidant activities. And also by the LCMS, we found such compounds like isolicuriteginine, some coumarin compounds, which are responsible for anti-inflammatory activities.
Yeah, but LCMS you cannot prove. There is no like data. Did you publish this data of LCMS results?
Not yet, but they are approved by the conferences, by the approbation at the international conferences. And it's planned to publish it regarding to my project.
Okay, thank you. That's all from my side.
Thank you. Thank you so much for your interesting questions.
Can I ask any question? Can I
Yes.
ask question? Any question? My question is, from what deposit was the bentonite clay used and why? Why
Thank you.
did you use this bentonite clay?
Thank you, Professor. This bentonite clay was collected from the Mandrak deposit. This deposit is rich for bentonite clay materials, and this bentonite clay was chosen because this bentonite clay has more than 70% of montmorillonite in the content, and this montmorillonite is good for using as a medical drug because it's easy to use, painless, and also good for use for human organism. It has a good biocompatibility.
And thank you. The second question, why did you choose cellulose for hydrogel synthesis? It is known other materials for hydrogel synthesis. For example, synthetic polymers, copolymers, and other compounds.
Hydroxypropylmethylcellulose, we made some literature review, and the hydroxypropylmethylcellulose is good candidate for using as a drug for hydrogels, to using as an ointment for transdermal materials, and it has a good property to absorb some biological active compounds itself, and to has a good prolonged release of the active ingredients. That's why hydroxypropylmethylcellulose was chosen, and also hydroxypropylcellulose is a known polymer, natural polymer, which is used at medicines. It can be capsules or films or gels, and it's already known as this polymer base.
Thank you.
Thank you so much.
Thank you, colleagues. I have several questions to RLM. When you obtained extracts on the base Tamarix hispida, you used ultrasonic methods of extraction, yeah?
Yes, yes.
Please tell me what the advantages of these methods when we comparised of traditional methods, and do you know what the traditional methods used when we obtaining extracts on the base Tamarix hispida, and when we in opinion safe of biological activity properties of Tamarix hispida.
Thank you for your questions. Yes, nowadays here a lot of types of extraction methods, but nowadays the ultrasonic treatment have a more popular, because by the ultrasonic treatment we can decrease the time of extraction, because some traditional extractions such as maceration method needed like 24 48 hours for extraction, and by using ultrasonic treatment we can decrease the time until, for example, 30 minutes, 40 minutes. Yeah, that's why here we use it first time for this plant ultrasonic treatment.
And when we use these methods, you saved biologically active properties of
Yeah,
extracts, yeah?
Yeah, sure. The biological
activity. Did you study?
Yes, this all biological activities is for the THU10 extracts. With for extracts which is obtained by the ultrasound method.
Thank you. And when you obtained hydrogel, you used several components, yes, methylcellulose extracts on the base Tamarix hispida. and bentonite clay, yeah? Okay, what's the molecular interaction between components of hydrogel determined of the stability and rheological properties of hydrogels?
Thank you for your
And
question.
how this influence on, for example, other properties of hydrogels?
Thank you so much for your question. Actually, it's a very important question because some chemical interactions between the HPMC, bentonite clay, and Tamaris hispida extract. We can. It seems to be that here some hydrogen bonds between them, hydroxyl group of the THU extract components and the bentonite clays, mineral oxide, some silicium oxide or aluminum oxide. Here are some hydrogen bonds, and also in HPMC, here we have methoxy and hydroxy and propyl groups. We can also see these bonds. And also here are some physical interactions like absorption on the surface and interlayer absorption of these extracts between the bentonite and the HPMC.
Okay, you in your annotation, you noticed about antioxidant activity and anti-inflammatory effects in vivo, okay? How this molecular interaction influence on these properties obtained hydrogels?
Yeah, I understand your question. Thank you. So we chose it 1% of THU extract and obtained the results of the kinetics of release. And we calculated the release of this extract THU10, and this release, that amount was enough for showing this antioxidant and anti-inflammatory activities.
Okay, and one questions in your annotation you noticed about you can create in the future smart hydrogels, yes, on the
Yeah.
base you obtained. Okay, what's did you have barrier to create these smart hydrogels now?
No, thank you for your questions. Maybe it can be like a recommendation from you. Thank you so much. Yeah, it's not a barrier. So now I have a project like Just Film, and also after this project, maybe if everything will be okay, maybe we can use some commercialization of these hydrogels. And thank you, yeah, for your recommendation. Maybe to have some smart hydrogels, we can also to calculate
what you will do to create these smart hydrogels on the base developed hydrogels.
I think it's ready to use, but just we need to commercialize these hydrogels.
Thank you.
Yes, I have one question. Arelum, in this table 3, you are showing anti-inflammatory activity of 3 extracts, but 70% extract is showing good well. Why you are not using for hydrogels, not 70% or 10%?
Thank you for your question. As we can see in the table, the solubility of THU70 extracts is partially, and it's not as good for using it as a biological active compound when we immobilizing them into the polymer carriers. So as we can see, THU10 also exhibits anti-inflammatory activity, and it's fully soluble in water and water solutions. That's why we use a THU10 as a biological active extract by using for carriers.
Okay, thank you. Dear colleagues, are there any questions to Арайлым? Құрметті әріптестер, қазақша, орысша, ағылшынша қоя берсеңіздер болады. Сұрақтарыңызды. Сұрақ жоқ болса, онда келесі сатыға көшейік. Are there no more questions? The floor is given to the scientific supervisors. Абилов Жарылқасын Абдуахитович, Doctor of Chemical Sciences, Professor of the Chemistry and Technology of Organic Substances, Natural Compounds and Polymers, Al-Farabi, Kazakh National University. Please.
Спросить, как себя чувствуешь? Мне хочу ужасно. Почему? Потому что, смотря на членов ученого совета сегодня, которые рассматривают работу моей аспирантки, тем, что давно, что мы не были на таких защитах, ну, скорее всего, наверное, не зовут присутствовать, но насколько изменилось все за то время, которое, в принципе, прошло. Почему? Потому что 1966 года, когда я прошел студентом, и по сегодняшний день я открывал и закрывал только двери химического факультета. И здесь, смотря на членов совета, слыша то, что задаются какие вопросы, и самое главное, что я здесь не услышал ни одного такого разговора, которые были бы через членов совета на казахском или русском языке. Потому что, ну, не все мы, наверное, понимаем. И это, в принципе, наверняка радует тем, что мы присутствуем на таких защитах, которые меняются вообще, как наш весь мир. Работа сегодня мы представлены на защиту сегодняшний день Арайлым. Это, ну, наверное, продолжение всех тех исследований, которые проводились на нашей кафедре. И тому, что уровень, который не упал, может быть, вот по химии природного соединения в особенности, это связано многим лицам, которые сегодня являются как членами совета, так и большими учеными. И в особенности то направление, которое происходит, изучать это солеустойчивые растения, которые имеются для Казахстана очень большое значение. И, конечно, сегодня можно было бы сказать большое спасибо Серғазы Мужсарычу, который, в принципе, эту работу, которая проводится у нас в Казахстане, и в основном, конечно же, в Караганде, но благодаря академику Серғазы Мужсарычу. Члены сегодняшнего работы, которые слышат сидящие здесь ученые с Турции, ученые из Пакистана, которых давно проводим такие работы в области химии природных соединений, это, конечно же, в принципе, радует. И я считаю, что Арайлым, работая в Пакистане, в университете имени Карачи, сделал большую, в принципе, работу. По крайней мере, нам это очень так, в принципе, кажется. И хотел бы я поблагодарить всех членов диссертационного совета, что все вы сегодня благожелательно прослушали доклад Арелем. Я надеюсь, что она оправдает ваше доверие и в дальнейшем будет продолжать работу в области солеустойчивых растений. Спасибо.
Рахмет, Жарылқасын Абдахитович. Пікіріңізге. Так, the next foreign supervisor Iqbal Chaudhary, Doctor, Professor, Coordinator, General Comstech, Distinguished National Professor, Honorary Advisor, ICCBS, Karachi, Pakistan. Dear Professor Iqbal Chaudhary, please.
He gave a note. Kindly read the note, please. In messages.
Okay. The floor is given to Scientific Secretary Bulat Uralbekov to present opinion of foreign supervisor Iqbal Chaudhary.
Yeah, dear colleagues, I have the original letter from Professor Iqbal and the reviewer. So I will not read all, but the only main issues. He concluded in his review that Amanzholkyzy RLM did her research as a collaborative student under his supervisions at the Hashni Ibrahim Jamal Research Institute of Chemistry, International Center for Chemical and Biological Science, University of Karachi. And during her PhD research, she worked on HPLC, LCMS, CM, and performed extraction, column chromatography, vacuum liquid chromatography, thin layer chromatography, and many others in vivo studies. As a result, she gained experience in column chromatography, compound isolation, identification of chemical compositions, and evaluation of biological activities. So in general, Amanzholkyzy can be described as a responsible young researcher, and so the thesis has great theoretical and practical significance, and he concluded that she carried out meets the requirements of the PhD dissertations, and therefore the Amanzholkyzy RLM deserves to be awarded the degree of doctoral philosophy, PhD, under the educational program chemistry. Professor Dr. Iqbal Chaudhary, Mustafa Prize Laureate.
Thank you. The next, the floor is given to the official reviewers. The floor is given to the first reviewer, Tatakhanova Gulnur Sayranovna, candidate of Chemical Sciences, Associated Professor, Deputy Head of the Laboratory of Engineering Profile of the Satpayev University, Almaty, Kazakhstan. Gulnur Sayranovna. Естіп тұрсыз ба? Естіліп тұр ма?
Yeah, естіп тұрмын.
Аха, сөз сізге.
Thank you. Review of the official review for dissertational work are Amanzholkyzy RLM on the same hydrogel forms of hydroxypropyl methyl cellulose and bentonite clay based on plant extract Tamarix hispida, presented for the degree of Doctor of Philosophy in the education program AD-5301 Chemistry. Criteria number 1. The thesis was carried out under the program Jas Ghalem, grant funding of the Community of Science of the Ministry of Science and of Higher Education of the Republic of Kazakhstan. Development of hydrogel forms of plant extract based on HPMC between 2024 and 2026 under the priority the scientists of life and health. Criteria number 2. The dissertation makes a significant contribution to the field of science, and its importance is thoroughly substantiated. The key foundation related to the development of hydrogel with tamarisk haspida extracts, the optimization of extraction yields, and the identification of patterns in the composed carrier formation effectively addresses a relevant research problem. The result advances our understanding of macromolecular and natural product chemistry, as well as the design principles of composed biomedical materials. Criteria number 3. The degree of the author independence is demonstrated through the critical analysis of scientific literature and preparation of the comprehensive review on hydrogel and polymer composite systems. In the independent exclusion of the experimental part and the subsequent interpretation and generalization of the research findings. The PhD student also showed her skills of experimental work on chromatograph, study of rheological characteristics, and carrying out analysis to establish antioxidant, anti-inflammatory activity. The author prepared materials for publication in scientific journals and presented them in the form of reports at scientific conferences. Criteria number 4. Justified. The literature review provides a solid rationale for the research direction, focusing on relevant areas such as cellulose, ether hydrogels, pharmaceutical applications, plant-based bioactive complex immobilization, and tamarisk haspida constituents. The content of the thesis reflects the topic of the thesis. The purpose and objectives fully correspond to the topic of the thesis, focusing on the solving the important problem of carrying phytoextracts with anti-inflammatory and antioxidant activities and polymeric hydrogel systems characterized by important physico-chemical properties. All sections and arguments of the thesis are complementary interconnected, and the conclusions accurately reflect in main results. The foundation logical flow for the experiments and analyses conducted align with the state goals and objectives. The dissertation demonstrates and critical analysis. The author proposed novel solutions to key objectives, particularly in HPMC gel formulation, composite development. The solutions are well reasoned, justified through comparative analysis with authoritative literature and informed by recent scientific advances in the relevant field. Criteria number 5. The dissertation put forward novel scientific findings, not previously dissertation in the scientific literature. In particular, this study in the first two demonstrated notable antioxidant and anti-inflammatory effect of tamarisk haspida aerial part extracts for was extraction and immobilization of the extract in HPMC and HPMC bentonite composite hydrogels. This research offers novel experimental results. The key innovations include, first, use of ultrasonic for enhancing active component extraction. 2. Identification of antioxidant and anti-inflammatory activities in the specific aqueous ethanol extracts. 3. Development and optimization of novel HPMC bentonite gels. The novelty is validated by a patent the Republic of Kazakhstan and Scopes indexed publications. The research foundations support novel and justified technical and technological solutions, including a gray ultrasonic extraction methods that improves BAS yield and bioactive yield, decreasing extraction time with novel gel developments. Criteria number 6. All main conclusions are based on scientifically significant evidence. The reliability of these conclusions is based on comprehensive experiments using spectrophotometric, rheological, and biomedical methods. The results are well interpreted and confirmed. 7. All three provisions presented in the dissertation work are logical work for the conducted research, are confirmed experimentally, and are not trivial. The novelty and practical significance of the provision are proved by publications in peer-reviewed journals and a patent for a utility model. Criteria number 8. The choice of research methodology is well justified. Methods and approaches are used based on the analysis of world practice. The author's analysis of approaches and methodology is given in the analytical literature review. The selected methods are used in the experimental part. In the dissertation work of the applicant, the scientific results of these were obtained using modern analytical and experimental approaches, including ultrasonic extraction, biological activity test of DPPH, analysis of rheological properties, desorption characteristics. These theoretical conclusions, models, identified relationships, and patterns are provided and confirmed by experimental research, which include instrumental study of isolate extracts, establishing the main characteristics of gels, conducting biotesting of extracts, and reviewing immobilization patterns. Important statement are confirmed on the basis of analysis of modern scientific achievements and confirmed by reference to current and reliable scientific literature. These theories used are published in peer-reviewed publications on scientometric data bases. Included works both on the chemical composition and biological activity of plant extracts and on hydrogel systems with plant components. The literature sources used are sufficient for the literature review, including a list of 193 titles from the reliable and relevant literature. Criteria number 9. This dissertation studies timely issues in the area of material science. The creation of composed hydrogels using grain methods and novel extraction techniques for Tamarix haspida water-soluble extraction further the underlying understanding in the field. This thesis is of particular importance and there is higher probability in the possibility of developing transformal forms with prolonged action, and proposal can be used in the pharmaceutical practice. Water-soluble extracts and gels based on HPMC and its clay composition are proposed for practical application, which can be used in medicine as agent with antioxidant and anti-inflammatory activities. Criteria number 10. The quality of academic writing is high. The text is structured, the presentation is logical, scientific terminology is used, the design complex with the requirement and includes tables, figures, the necessary list of sources of literature. Criteria number 11, notes on a thesis. First, on page 38, Formula 3 might contain an error. The factor of 100 is likely a typographical error. 2. In the dissertation, figure 89 presents the effect of temperature on the swelling kinetics of bentonite clay with THU10. Judging from the graph, these are result of studying the swelling kinetics. Nevertheless, its remains unclear whether separate studies were conducted on the effect of temperature directly on both swelling kinetics and drug release, and if so, at what temperature? 3. If such separate studies were conducted, what were the main results? The lack of detailed information in the description of figure 89 makes it difficult to fully understand this part of the work. 4. The dissertation details the release of THU10 extract for the hydrogel, but the method of extract immobilization with the hydrogel is not described. What method was used to immobilize the extract into the hydrogel? 5. If the extract was incorporated during synthesis, were the extracts prepared assessed after exposure to the 8 degrees of Celsius system's temperature, or synthesis temperature? Criteria number 12. The candidate has demonstrated research productivity through publications in high-quality journals. Moleculars Q1, a utility model patent, and presentations at international conferences. Those achievements confirmed the candidate's scientific maturity and the significance of the dissertation topic. Amanzholkyzy Araylim's dissertation prepared on the topic hydrogel forms of hydroxypropyl methylcellulose and bentonite clay based on plant extract Tamarix haspida is submitted for the degree of Doctor of Philosophy in the specialty 8D05301 chemistry. In the terms of relevance, scientific novelty, significance for theory and practice, and the scope of experimental research, the dissertation fully meets the requirements for dissertations for the degree of Doctor of Philosophy in the specialty chemistry. The official description of the reviewer is that Amanzholkyzy Araylim deserved to the award the degree of Doctor of Philosophy in the educational program chemistry. I'm finished.
Okay, thank you, Gulnur Sayranovna, for your details review. The floor is then given to the applicant, Araylim, to give some comments to review.
Thank you so much for your review. And by answering for your notes, first, here's some typographical error as I can show in the presentation. Here are some typographic error, and we just forgot to mention the temperatures in the graph. As we can see in the graph here, the increase in the temperature, we can see the increase of swelling degree of the bentonite clay. And yes, we I can show in the presentation. If it's not open, I can explain that the graph shows the swelling kinetics of the bentonite clay. Oh, just a moment. Yeah, here, just this line of words was typographically error. And here we can see by increasing the temperature of them, by the increasing of the temperature, the swelling kinetics of bentonite clay also increases because of the hydrogen's bonds by the increasing the temperature weakening, and that's why the temperature also affects for the swelling kinetics. This is the answer for first and second question. And yes, the lack of the third question, the lack of detailed information in the dissertation work is forgotten, but I hope this mistake is not affect to the final decision. And the fourth question was how the THU extract is immobilized to the polymer base. So as we know, the extracts higher than 40 degrees can be disturbed because of here we have a lot of organic compounds which is spoiled at the high temperatures. So firstly, one part of the cold water dissolved THU extract, and HPMC dissolved at 1680 degrees. That's why it's a hot solution. So we are waiting for decreasing the temperature and then adding this extract and then mixing them together at the stirrer. Magnetic stirrer. That's why the compounds in the extract cannot be spoiled by the method. Thank you.
Thank you, Aylem. I am agree.
Gulnur Sayranovna, are you satisfied of answers? Aylem?
Yes, yes, I am satisfied.
Okay, thank you. Thank you, Aylem. The floor is given to the second review. Tajbayev Yerkebulan, Doctor Chemical Sciences, PhD, Professor of Buketov Karaganda University, Karaganda, Kazakhstan. Dear colleagues, меня слышно было, да? Құрметті рецензенттер, кетіп қалмаңыз, жарай ма? Соңына дейін болу керексіңдер.
Жақсы, бастай бере ма?
Иә, бастай беріңіз.
Official review, dissertation, hydrogel forms of hydroxypropyl methyl cellulose and bentonite clay based on plant extract Tamarix hespida. Candidate Amanzholkyzy RLM, specialty chemistry. Dear chair, members of dissertation council and colleagues, my detailed written review has been submitted to the dissertation council and is available on the official website. Therefore, in my oral presentation today, I will briefly highlight only the main point of the review. The dissertation was carried out within the life and health sciences priority areas under the Jazz Galim grant program and addresses on important and timely problem, the development of biodegradable and biocompatible hydrogel carriers for prolonged drug delivery system. Aim and scope. The work is devoted and create hydrogel system based on HPMC and bentonite clay with the immobilized plant extract Tamarix hespida. The candidate perform extraction in identification and of activity compounds, biological activity studies and details, physical, chemical, and rheological analysis of obtained hydrogel. Remark figure 10 of 36, which show the UV spectra of aqueous solution of Tamarix hespida extra is very unclear. It is difficult to see at which wavelength the Tamarix maxima and upset. Second, the methodology of study drug release from the gel is based on incubate in aqueous medium or in UV spectrophotometry analysis. This approach is acceptable for preliminary evolution of release kinetics. However, the work lacks interpretation of the obtaining data using kinetics model, which would allow conclusion about the mechanism of drug release. Conclusion. The dissertation represents of complete and independent scientific study conducted high methodology and analysis level. I recommended awarding Amanzholkyzy Aralim the academic degree of Doctor of Philosophy, PhD in chemistry. Thank you.
Thank you, Dr. Erkibulan, for reviews. The floor is given to the applicant, Aralim, to respond to the reviews comments.
Thank you so much for your thorough review, and let me answer to your questions. In the figure 10 on the page 36, here some UV spectra is not clear. Yeah, we are sorry, but we used this UV spectrum for gallic acid intensity, and this spectrum is at the 268 nanometers, used as a standard for release kinetics of the tamaris hispida from the base. The second question, yes, it's very important to know the evaluation of the release kinetics, but in our work, this evaluation is not was a task. So that's why we didn't evaluate this parameter. But as we know that the release kinetics of the biological active compounds, some drugs can be evaluated by the Kosmeyer-Peppers models and Higuchi models, but usually these models are used for individual compounds, and here we have some biological active complexes. That's why this was not the task of our work. Thank you so much.
Thank you, Aralim. Dr. Erkibulan, are you satisfied?
I'm satisfied, yes.
Okay, thank you. The next is a discussion, and you have opportunity to tell your opinion about dissertation work of Aralim. Құрметті әріптестер, біз келесі жұмысымыздың сатысы жұмысты талқылау, қазақша, орысша, ағылшынша өз пікірлеріңізді диссертациялық жұмыс бойынша айтсаңыздар болады.
Can I express my opinion about this dissertation work?
Please.
Thank you. I think today we have listened to very interesting dissertation work. This dissertation work is devoted to the production of medicinal substances, materials based on domestic raw materials. It is very important for our country to use domestic raw materials to produce drugs. I think the results will contribute to the development of this very important scientific field, and the results have been published in highly regarded journals, and in my opinion, the dissertation work meets all the requirements. So I support this dissertation work and believe that Aralim deserves to be awarded the degree Doctor of Philosophy. Thank you.
Thank you, Dr. Abdiyev. Please, dear colleagues, you have opportunity to share your opinion.
Yes, if possible, I can share, Dr. Janar.
Yes. Please.
Okay, thank you, Aray, for to deliver your PhD thesis. I have well assessed the PhD thesis, which is a thorough exploration of academic and the notable economic significance, according to me. The thesis outlines the materials and the methods employed throughout the study, where systematic approaches were applied and to ensure accuracy, scientific rigor across experimental and clinical phases. All procedures are, according to me, explained, emphasizing the authenticity of the work. The findings are clearly outlined in the thesis. Tables and figures provide a self-explanatory insights, and the results are critically compared with the other studies. Some parts of the PhD thesis published already. Finally, all findings are summarized in the thesis conclusion. As a summary, the thesis is both well written, throughout produced. The research project was carefully explained. Experiments are conducted with a precision, data were analyzed effectively, and the results were displayed in the tables and graphs. Furthermore, the data were presented and discussed scientifically. Additionally, the research topic is current, relevant, and high significant worldwide, including, of course, Kazakhstan. Therefore, I also recommended without any hesitation that the candidate arrive should be awarded the PhD degree with this study. Thank you so much.
Thank you, Dr. Öztürk.
Рұқсат па екен?
Иә, әрине, рұқсат.
Рахмет. Мен енді Жарылқасын Абдуахитұлын қолдап орысша айтайын, ағылшынша да, қазақша да айтуға болар еді, бірақ Жарылқасын Абдуахитовичті қолдап орыс тілінде пікірімді айтайын. Во-первых, хочу поздравить профессоров Жарылқасын Абдуахитовича Абилова и Мухаммад Игбал Чоудхари с очередным научным достижением. Это хорошей, содержательной работой, диссертационной работой Арайлым Аманзholkyzy, которую мы здесь внимательно прослушали с вами. Отличительной особенностью и основными преимуществами диссертационной работы Арайлым Аманзholkyzy это прежде всего определение оптимальных условий ультразвуковой экстракции сырья тамарикса хиспида. Это с использованием воды и этанола для повышения выхода суммы экстрактивных биологически активных веществ. Это прежде всего уже работа считается готовой к технологии, к фармацевтическому производству, потому что использование ультразвуковой экстракции и соотношение воды с этанолом соответствующим, это, конечно, привлекательно для технологов. Во-вторых, определена, как вы обратили внимание, выраженная противовоспалительная, антиоксидантная активность полученного экстракта из тамарикса. Тоже является хорошим вкладом данной диссертационной работы. И в-третьих, следует отметить, что разработан оптимальный состав трансдермального гидрогеля на основе бентонитовой глины, гидроксипропилметилцеллюлозы и экстрактивных веществ тамарикса-хиспиды. Здесь хочу обратить внимание, что на фармацевтическом рынке вообще, уважаемые коллеги, известны такие препараты на основе бентонита, которые мы с вами хорошо знаем. Прежде всего, адсорбирующий противодиарейный порошок Смекта. Смекту мы все хорошо знаем. Во-вторых, это лечебно-профилактическая зубная паста Бента. На основе бентонитовой глины для зубной пасты Бента впервые было разработано профессором Сахиповой Зурьяды Бектимировны. Она защищала докторскую диссертацию на ученую степень доктора фармацевтических наук в 2010 году. Эта работа тоже очень прекрасная, и Бента сейчас хорошо используется на практике. В-третьих, это зарегистрированные и используемые тоже в фармацевтическом рынке противовоспалительная мазь Метробент. В сравнительно представленных лекарственных препаратов, которые зарегистрированы в Казахстане, отличительная особенность работы Арайлым – это разработка, впервые разработан в трансдермальной форме, и при этом использована гидрогелевая форма бентонита, которая включает гидроксипропилметилцеллюлозу и биологически активные компоненты тамарикса-хиспиды. Тамарикса-хиспиды состав очень интересный в плане полифенольных соединений и ряда других компонентов. Они хорошо изучены в работе доктора химических наук Султановой Нургуль Адайбаевны, также ученицы уважаемого Жарылкасына Абдуахитовича Адилова. Эту работу, которая была прекрасно тоже защищена, мы были свидетелями как раз защиты данной работы профессором Султановой в 2010 году. Поэтому достижение и достоинство работы Арайлым Аманзholkyzy, что она продолжила эту работу и довела до практического внедрения технологии фармацевтического производства в виде уже трансдермального лекарственной формы, разработанной на основе бентонитовой глины гидрогелем. Таким образом, диссертационная работа Арайлым Аманзholkyzy представляет собой целостные, завершенные научные исследования, проникнутые внутренним единством, и результаты, полученные автором, обладают, несомненно, новизной, фундаментальной, практической значимостью. И все вышеизложенное позволяет мне сделать заключение, что Арайлым Аманзholkyzy вполне заслуживает присуждения искомой ученой степени доктора PhD по специальности химия. Назарларыңызға рахмет.
Көп рахмет, профессор Адикенов. Тағы пікірлеріңіз болса, бөлсеңіз болады. Булат Нурланович, пожалуйста, please.
Спасибо. Коллеги, я просто хотел отметить касательно метода, то, что вопросы я задавал. Да, к сожалению, мы уже второй раз сталкиваемся с кафедры. I will be in English, okay, because the difference is in English. So that's the second time actually when we have this problem with the Department of Organic Chemistry with the methods based on mass spectrometry. So unfortunately, the PhD candidates cannot respond to the questions about the method, about the equipment and specific type of ionization, detection, and the structure. Yeah. In my opinion, so all the data which was presented in the dissertation, so the student should know it and understand the principle of the method and defend these results. Because they are important, especially on the identification of the compounds which are present in the sample. Yeah, so in my opinion, these results, so I see no evidence in the dissertation. I see only molecular mass and that's all. Like M to Z ratio, no spectra provided, no chromatograms, no equipment type, parameters of detection, like type of mass analyzer. So this important information is missing. Yeah, so these methods are very important in my case, in my view, in the dissertation. It's they are not directly connected to the provisions in the dissertation, but anyway, this is I think important part of the dissertation. So and I ask for the future dissertations which will be presented to pay like big attention to this issues with the methodology. Yeah, and I wanted to ask foreign supervisor about this analysis, but unfortunately he was not reporting, Professor Chowdhury. So last time at the defense I asked some questions, but this time unfortunately I couldn't ask. Yeah, maybe he can clarify some issues, but unfortunately not. So that's why I'm still not convinced in these results on the identification. Yeah. Okay, that's all from me. Thank you.
Булат Нурланович, вы закончили, да?
Да, и все. Спасибо.
А то перебили, ага. Спасибо. Thank you. Your opinion. Do you have?
Құрметті әріптестер, енді мына не бойынша қазақша айта берейін мен. Бұл жұмыс енді біздің кафедрада басталған жұмыстардың бірі. Соның жалғасуы әрине профессор Жарылқасын Абдуахичтің бастауымен оның шәкірттерімен көптеген жұмыстар жасалды. Соның ішінде бентанитсазмен және тамарикс хиспида, бұл жыңғыл өсімдігінің бір түрі, өте биологиялық белсенді заттарға бай болғандықтан оны әрі қарай әрине бентанитсазмен және гидрогелмен біріктіріп басқа да дәрілік заттарды алуға жұмыстанды. Арайлымның жұмысы бүгінгі тыңдадық. Қызықты жұмыстардың бірі және өз деңгейінде жасалды деп ойлаймын. Және кішкене мана LCMS не бойынша әрине ол обычно HG'ді біз лабораторияға мана тапсырамыз образецті сэмплді. Ол жердегі специалистер өздері жасап, бізге тек қана анализді береді. Сондай да жағдай болған шығар деп ойлаймын. Енді профессор Игбалмен артынан түсіндіре жатыр Арайлым. Бірақ жалпы бұл жұмыстың несі де жалпы жұмысқа қойған мақсаттар және ашылды деп ойлаймын. Және алға қойған мақсаттарға жетті деп ойлаймын. Рахмет.
Рахмет, Балағыз Қамзғалиевна. Тағы да пікірлеріңіз бар ма? Құрметті әріптестер, диссертациялық кеңестің мүшелері. Dear colleagues, do you have opinion? Okay. A break is announced for the secret ballot.
Yeah, dear colleagues, just a moment. I will move the not members and not reviewers to another hall. So please stay just a few minutes.
кешіріңіздер, біз қалай дауыс береміз? Почта арқылы ма или телефон арқылы ма?
Қазір экранда шығады сізге. Құрметті әріптестер, қазір сізде can see on the monitor, and you have an opportunity to give voice on the monitor, by the monitor.
Раз. So, dear Professor Surk, just on the monitor. Уважаемые коллеги, сейчас на вашем мониторе появится, как его, опрос, только один выберите, и все на это.
На экране надо отметить, да?
Булат Муратович, это не тот опрос, это для серии статей. Коллеги, слышно?
Dear colleagues, you have to choose only one clearance.
Булат Миланович, вы спрашиваете, да, что-то?
Ну, это неверный вариант. Должно быть 4 варианта. У нас же не серия статей.
Это неверный вариант. Я вот смотрю, что это неверный вариант. Сейчас, подождите.
Да,
тогда...
там есть именно трехязычный, по-моему. Голосовать, да, не в избранном, а в обычных.
Все понял.
Где 4 варианта. Там казахский, русский, английский.
Yeah. Я что-то не могу здесь разобраться сейчас.
Ну, или можно этот вариант поправить, просто добавить один вариант туда. Там есть редактировать.
Здесь просто выбор наилучшего подхода, secondary poll, голосование после... Видите? То есть... Все хорошо будет. Да. Как его добавлять-то? Тогда я, наверное, вас сделаю, да, организатором. Попробуйте сделать. Что-то я не могу добавить.
Я сделал себя.
Ага.
Сейчас.
Dear colleagues, you have to give your voice again. Құрметті әріптестер, дауыс беруіңізді сұраймыз.
Сейчас, подождите, уважаемые коллеги, у нас немножко техническое это. Сейчас мы перезапустим опрос. Так, Булат Иванович, если вы будете запускать, тогда я из одного выйду.
Да, тогда я со второго подключу сейчас. Или нет, я сейчас верну вам, может быть.
Хорошо, хорошо.
Хорошо, не выходите. Ага.
Так, уважаемые коллеги, я запускаю опрос
Да, там в избранном опрос этот.
в избранном.
Dear colleagues, we have give yours again, please.
Ну, по идее, 9 должно быть, да? Уважаемые коллеги, надо проголосовать еще один. Все. Результаты готовы. Да, завершить опрос сделаю, да? Сейчас я всех сейчас приглашу, потом хорошо.
I ask the members of the dissertation council to approve the voting protocol. And based on the defense and the results of the secret online voting, the dissertation council at Al-Farabi Kazakh National University award the degree of Doctor Philosophy, PhD. Amanzholkyzy Araylim for the dissertation on the topic hydrogen forms of hydroxy propyl methylcellulose and bentonite clay based on plant extract Tamarix hispida specialty AD05301 chemistry.
So, congratulations.
Құрметті әріптестер, қорғауды аяқтауға рұқсат беріңіздер. Арайлым Аманжолқызы, құтты болсын, Жарқасын Адахитович. The next word to Araylim, please.
Thank you so much. And I also want to say my thanks for my foreign supervisor, for Dr. Professor Muhammad Iqbal Chowdhury, for his support and giving opportunities to work in high-level laboratories. Также хочу выразить большую благодарность своему руководителю Абилу Жархасну Дахитовичу за его поддержку, за наставничество не только в науке, но и по жизни. Также хотела бы поблагодарить свою семью, родителей, братишку, сестренку за такую поддержку и за то, что привили любовь к науке еще с детства и делали все возможное для этого. Также хочу поблагодарить кафедру, факультет химии, а также коллег, друзей за всю поддержку и любовь, которую проявляете. А также хотела бы выразить благодарность супругу и его семье. Я чувствую вашу поддержку и любовь. Спасибо вам большое. Также хотела бы поблагодарить диссертационный совет и рецензентов за оценку работы и за возможность выступить в диссертационном совете. Спасибо большое.
Спасибо, Рахмет. Thank you all for participating in the defense.
Yeah, so dear colleagues, thank you, Рахмет, Куб, Рахмет, что уделили внимание. Надеемся увидеть вас скоро на других защитах. Спасибо, что уделили внимание.
Рахмет, көп-көп, сау болыңыздар. Надеемся на дальнейшее сотрудничество.
Жақсы, сау болыңыздар.
Жақсы сау болыңыздар. Құттықтаймыз.
Sağ olun.
Kutlarım Aray
Sağ
seni.
olun.
Rahmet,
Rahmet.
teşekkürler.
Sağ olun.
Thank you. Teşekkürler.